Every cell in your body has a physical representation of that genome inside the cell and has to deal with it. In order to read it, to work with it, it has to physically operate that molecule.
Just the idea that a 2-meter long DNA can be wrapped up in a tiny, tiny cell is just mind-boggling.
So just imagine putting a whole football field inside a small backpack. The amount of compaction you need to do is what happens when you have the 2-meter long chromatin folded inside the nucleus.
And it isn't just randomly thrown in there like a bowl of spaghetti. In fact, it's a really highly organized structure.
The 4D nucleome represents how the linear genome is folded in the 3-dimensional nucleus, not only in the X, Y, and Z directions, but also over time.
The way that the DNA is organized in the cell actually has a lot to do with how a cell functions.
Being able to know that interaction and relationships helps us understand how error in those things can lead to developmental issues or even diseases.
ForDN started about 10 years ago. At that time, the question of what the human genome is folded like in cells was well known, but very few answers were available.
Rather than individual researchers doing their little own thing, we thought, why don't we bring people together, think of a central set of questions, get the best technology available, develop newer ones, and put the data together to learn something as a whole.
I think the program went a long way towards answering some of these questions. We now have started to understand some of the molecular mechanisms by which cells fold their chromosomes. And that, I think, is really a major breakthrough. I'm not saying we have now answered all the questions, but we are now in a position where we weren't 10 years ago, that we can start to apply these methods and these insights to try to understand how the human genome is regulated and how it goes wrong in disease.
Together as a community, we have discovered multiple important cases where the 3D genome will be disrupted, and that will be a causal contributor to either the onset or the progression of a human disease. Our own work has been in both Alzheimer's as well as Fragile X syndrome, but there are many other colleagues that have shown a link to genome miswiring and disease acquisition.
There have been some paradigm shifts, actually, within this program, where people did not think about how the chromatin folding is really related to disease biology. Now we have started to see that.
The technology that's involved in profiling the 3D structure of DNA has really just gone through a tremendous change over the last decade or so.
Those tools were not there before. And so we very much had to really develop these tools to study chromatin folding.
We now have to our availability a large set of methodologies, both molecular, genomic, imaging, but also computational methods.
And we can do things that we just could not dream of 10 years ago.
In this case, each technology that was developed opened up an entirely new capability. Technology has allowed us to see structures that were not seen prior to this, to map them in any cell type, and then begin to do experiments and then assess how structure gives rise to function in the case of the genome.
With 4DN, we were funded to build a new tool that we developed called Sprite. And we've actually been able to make this accessible to the community and broader field.
The sheer amount of data that we have generated as part of this consortium is a widely used resource for the community. The data portal that we built is being accessed by 50 countries. Thousands of, maybe tens of thousands of people all around the world are accessing the data sets that we generated. Everything is documented. All the intermediate steps are available, and so anybody, they can all come and look at this data very easily.
All of us have been committed to developing state-of-the-art imaging and genomics technologies, but not just hoarding them for ourselves, but rather providing freely protocols and code as well as sort of a community of people willing to help each other to adapt the newest technology.
To study the human genome, it needs to bring together people from very different disciplines. And that is what happened in this consortium.
This is a consortium that's funded to generate data for the community, which means we should release the data as quickly as possible. And we did something that others have thought about but could not do, which is to make everything completely open.
This is the first major biology consortia to adopt the preprint publication policy. That led to a better understanding and trust among colleagues.
And I think it really created a very collaborative and transparent culture. And it just changed the way how we do science. For the nucleome research in particular, it can only be done when people work together. And that has happened in this consortium. And I look back and I'm kind of amazed how that worked.
You can see that there's really great collaboration between people, and ideas are being shared around, and people are being able to contribute together.
They are exchanging ideas, they are talking to each other, and therefore that is, to me, a good legacy.
You know, many of the intangible values are the community that we created. The consortium embraced me from every angle and brought me in to interact with some of the best thinkers in our community as well as some of the best leaders. My PhD advisor, Victor Courses and Job Decker, were involved in this program from the very beginning, and now I'm coming through the ranks. My trainees are, leading some of the sessions. Now at the end of these 10 years, I'm standing right next to my postdoc mentor. It's been quite rewarding, I think, to see the generations come through this program and to continue to serve the younger trainees that are coming through.
The type of people you interact with and mentor you really sets the tone for how you're going to feel for the rest of your career. So I felt familiar and supported. I became convinced that research was like an integral part of what I want to do as a career.
The 4DN project has actually really been fully transformative to my career. So it's really put me into a community of scientists that I'm really excited to be a part of.
4D Nucleome is going to be one of the most successful Common Fund programs at NIH.